Right now, as you are listening to this, deep inside the walls of your aorta, there are white blood cells that are literally eating themselves to death.
Yeah, it's pretty grim.
Right. They are just gorging on these microscopic droplets of toxic oxidized fat. And they keep eating, unable to stop until they just swell up, turn into this bubbling foam, and die, spilling all their toxic contents and building a microscopic graveyard right there inside your artery walls.
It is a brutal, microscopic battleground. And for most people, it's happening in complete silence, decades before a single symptom ever even appears.
Welcome to the Deep Dive. We are talking directly to you today. If you're prepping for your major pathology seminar, you know that just like memorizing a list of diseases is a total recipe for disaster.
Oh, absolutely. It won't work.
Right. You don't just need to know what these diseases are. You need to understand the biological crime scenes they leave behind. You need to know the why and the how so deeply that the histology slides practically read themselves for you.
Because pathology isn't just, stamp collecting. It's structural logic.
Yeah.
If you understand the normal blueprint of the human vascular system, and you understand the exact mechanisms of how that blueprint gets injured, the pathological outcomes, so the aneurysms, the plaques, the dissections, they all become entirely predictable.
Exactly. So our mission today is absolute mastery of this material. We are taking a microscopic, step-by-step journey through the plumbing of the human body. We're going to decode the normal structure, the precise cellular mechanisms of injury, the histology you are going to stare at under the microscope.
Which is huge for the exam.
Huge. Plus, the integration with immunology and the clinical logic for every single vascular disease in our source text. We are leaving no stone unturned.
So to understand the diseases that destroy the vascular system, we really first have to establish the baseline. We need to look at the normal blueprint, started with the hierarchy of the vessels themselves.
Okay, let's unpack this. Let's start with the arterioles. These are the small arteries, usually measuring anywhere from, 20 to 100 micrometers in diameter.
Yeah, right around there. But their size is incredibly deceiving.
How so?
Well, arterioles are the undisputed masters of your systemic blood pressure. And that authority comes directly from their architecture. If you look at an arteriole under a microscope, you'll see that its middle layer, the media, is composed almost entirely of tightly tacked, smooth muscle cells.
And those exist to do just one thing, right?
Exactly. They contract to cause vasoconstriction, or they relax to cause vasodilation. That's it.
And the source text gives us a mathematical rule that explains exactly why a tiny contraction in these microscopic vessels has such a profound systemic effect on the whole body. It's all about fluid dynamics. The resistance to fluid flow in a tube is inversely proportional to the fourth power of the diameter.
Right, that is Poiseuille's law, and it is the absolute key to understanding hemodynamics. It means the relationship isn't linear. So if the smooth muscle cells in an arterial contract and just halve the diameter of the vessel, the resistance to blood flow doesn't just double.
Because 2 to the fourth power is 16.
Exactly. You increase the resistance 16-fold with just a microscopic tightening of the vessel wall.
Wow. So if the heart is the pump pushing the water, the arterioles are like the adjustable nozzles on millions of tiny garden hoses scattered throughout your tissues, a microscopic twist of those nozzles, and the pressure inside the entire plumbing system just skyrockets.
That's a perfect way to visualize it.
Yeah.
And once the blood pushes past those high-resistance nozzles, it flows into the capillary beds. These are the exchange vessels, and their architecture is completely different. They're incredibly small, typically just 7 to 8 micrometers in diameter.
Which is fascinating because a normal red blood cell is about 8 micrometers wide.
Yeah, the red blood cells literally have to fold themselves.
We really, bend in half.
Yeah, they fold and squeeze through single file, and this is by design. It forces the cell membrane of the red blood cell into direct physical contact with the capillary wall, which minimizes the diffusion distance for oxygen and carbon dioxide.
Oh, that makes total sense. And to make that diffusion even easier, the capillary wall is stripped down to the absolute bare minimum. There's no smooth muscle here at all. It's just a single layer of endothelial cells resting on a basement membrane.
Right, along with a network of support cells called pericytes wrapping around them.
Pericytes, what do those do?
They're crucial because they help regulate endothelial cell function, their survival, and even the sprouting of new capillaries. But the most important hemodynamic concept for your seminar regarding capillaries isn't just their thin walls, it's their total cross-sectional area.
Wait, let me make sure I'm picturing this right for the listener. A single capillary is tiny, but you have billions of them operating in parallel.
Exactly. Think of a rushing, narrow mountain river that's your arterial system. suddenly emptying into a vast, incredibly wide, shallow lake. What happens to the speed of the water?
It drops to an absolute crawl.
Right. The volume of water is the same, but because it's spread out over such a massive area, the velocity just plummets.
That is the genius of the capillary bed. The massive total cross-sectional area drops the velocity of the blood to a near standstill. So this slow, lingering flow, combined with the single-cell thick walls, provides the perfect environment for the rapid exchange of oxygen, nutrients, and cellular waste between the blood and the tissues.
Exactly. The blood drops off the groceries, picks up the trash, and then it needs to head back to the heart. So it exits the capillary bed and enters the postcapillary venules. And this specific anatomical location is a massive high-yield target for pathology exams.
It is the absolute epicenter of acute inflammation, right?
100%. When you study vascular leakage, the fluid seeping into tissues to cause edema, or when you study leukocyte emigration, where white blood cells physically squeeze out of the blood vessels to hunt down a bacterial infection.
You must know that it happens almost exclusively in the postcapillary venules, not the arteries, not the capillaries, but specifically the postcapillary venules. But why there? Why that specific spot?
Two main reasons. First, the hemodynamics. The blood is just starting to gather speed again after the capillary bed, so the flow is still relatively slow, which allows white blood cells to sort of roll and adhere to the walls.
Okay, that makes sense. What's the second reason?
Second, the endothelial cells in the postcapillary venules are uniquely responsive to inflammatory mediators like histamine. When histamine binds to them, these specific endothelial cells contract. They physically pull apart, opening up gaps between themselves so fluid and immune cells can escape into the tissue.
So the venules are basically the exit doors for the immune system. From there, the blood flows into the larger veins to be carried back to the heart. And the veins have a completely different job description than the arteries.
Yeah, veins are capacitance vessels. They are the storage units. Compared to arteries, veins have much larger diameters, larger lumens, and significantly thinner walls with way less smooth muscle.
Because they operate under incredibly low pressure, they don't need to withstand the pounding force of the left ventricle.
Exactly. Because their walls are so thin and distensible, they can stretch to hold massive volumes of fluid. At any given moment, under normal resting conditions, approximately 2/3 of your entire body's blood volume is just passively hanging out in your venous system.
2/3. That's wild.
Yeah.
Okay, before we wrap up the structural blueprint, we have to mention the parallel plumbing system, the lymphatics. They are so easy to overlook, but clinically vital.
Yeah, the lymphatic system is essentially the body's secondary drainage and surveillance network. They're these thin-walled endothelium-lined channels that serve two main purposes. First, no matter how perfectly balanced your capillary exchange is, a little bit of fluid always gets left behind in the interstitial tissue space.
The lymphatics are the vacuum cleaner.
Exactly. They vacuum up this excess fluid and eventually dump it back into the venous bloodstream, primarily via the thoracic duct in the chest.
And if the lymphatics get blocked, say, by a tumor or a parasitic infection, that fluid builds up, causing massive lymphedema.
Right. And the second purpose is immunological. Lymphatics aren't just passive drains, they are highways for the immune system. They constantly transport antigen-presenting cells from their peripheral tissues straight to the lymph nodes, acting as a continuous surveillance mechanism, monitoring your entire body for invading pathogens.
Okay, we have the structural blueprint. We have the pipes. Now we need to look at the pressure inside those pipes and the incredibly complex biological thermostat the body uses to control it. Let's talk about blood pressure regulation.
The foundational physiological equation you need to know here is simple. Blood pressure equals cardiac output multiplied by peripheral resistance.
And cardiac output is just heart rate multiplied by stroke volume. So how fast the pump is beating and how much volume it pushes with each beat.
Right. So if the body needs to raise blood pressure, it essentially has two main dials it can turn. It can change the output of the pump in the heart, or it can crank up the peripheral resistance by contracting those tiny arterioles we discussed earlier.
And the master control board operating both of those dials is the RAAS system, the renin-angiotensin-aldosterone system. This is guaranteed to be a major focus of your seminar, so let's break this cascade down step by microscopic step.
The cascade begins in the kidneys. The kidneys are obsessed with maintaining adequate blood flow, because if flow drops, they can't filter toxins. So, located right where the arterioles enter the kidney's filtering units are these specialized cells called juxtaglomerular cells.
You can think of them as the body's ultimate, highly sensitive pressure and sodium sensors, right?
Precisely. They are constantly tasting the blood, feeling the pressure.
So what happens when the blood pressure drops? Or if the sodium concentration in the blood falls too low.
The juxtaglomerular cells instantly react by releasing an enzyme called renin directly into the bloodstream. Renin is the spark that lights the fire.
But renin itself doesn't raise blood pressure, right? It's just a messenger.
Correct. Renin is a highly specific protease, an enzyme that cuts other proteins. As it floats through the blood, it seeks out a large, inactive precursor protein called angiotensinogen.
Which is constantly being produced and released into the blood by the liver.
Yes. When renin finds angiotensinogen, it chemically cleaves it. It chops off a piece to create a new molecule called angiotensin I.
But the cascade isn't finished. Because angiotensin I is still basically biologically inactive, it's just a stepping stone.
Exactly. The blood carries this inactive angiotensin I out of the liver through the right side of the heart and pumps it into the massive capillary beds of the lungs. The endothelial cells lining the lung capillaries are studded with another enzyme called ACE, angiotensin converting enzyme.
Oh, wait, this is the enzyme that ACE inhibitor drugs like lisinopril are designed to block, right?
That's exactly right. As the angiotensin I flows past these lung endothelial cells, ACE grabs it and snips off 2 more amino acids. This final cut transforms the inactive molecule into angiotensin II. And angiotensin II is an absolute powerhouse.
Okay, listener, this is where you need to lock in the mechanism for the exam. Angiotensin II acts on 3 distinct targets to aggressively drive up blood pressure. Right, let's go through them. First, it attacks the peripheral resistance style.
Yes, it binds directly to receptors on the smooth muscle cells of the arterioles all over the body, causing them to violently contract. That intense systemic vasoconstriction causes an immediate, massive spike in peripheral resistance.
But angiotensin II also knows that to maintain that pressure long term, the body needs more fluid volume.
Right, so target number 2 goes straight back to the kidneys. It acts directly on the kidney tubules, forcing them to increase the resorption of sodium back into the blood. And the golden rule of renal physiology is...
Where sodium goes, water follows. By holding on to sodium, the kidney holds on to water, expanding the total blood volume.
Exactly, and it creates a backup system for this volume expansion with target number 3. Angiotensin II travels to the adrenal glands, the little triangular glands sitting on top of the kidneys, and stimulates the adrenal cortex to release a steroid hormone called aldosterone.
Aldosterone is the final enforcer. It travels down to the very end of the kidney's filtering system, the distal tubules and collecting ducts. And its only job is to upregulate highly specific sodium channels called ENaC channels.
By inserting more ENaC channels into the tubular membrane, aldosterone forces the kidney to aggressively pull even more sodium out of the urine and dump it back into the blood, dragging massive amounts of water with it. So the blood volume expands, the arterioles are clamped down tight, and the systemic blood pressure soars.
It is a brilliantly coordinated, multi-organ assault on low blood pressure. The kidney, the liver, the lungs, and the adrenal glands all working together. But if the system is that powerful, what stops it from blowing the pipes apart? How does the body counter-regulate if the RAAS system gets too aggressive?
The body uses the heart itself as a pressure relief valve. The heart isn't just a mechanical pump, it's an endocrine organ. If the blood volume gets too high, the muscular walls of the heart's chambers physically stretch beyond their normal limits.
The cardiac cells can actually feel themselves being pulled apart by the fluid volume.
Yeah, they do. And when they feel that excessive stretch, the cells in the atria release atrial natriuretic peptide, or ANP, and the cells in the ventricles release brain natriuretic peptide, BNP.
So these peptides are the biological brakes.
Precisely. They travel to the kidneys and essentially block the actions of the RAAS system. They force the kidneys to dump sodium and water into the urine processes called natriuresis and diuresis, and they also induce systemic vasodilation to relax the arterioles.
So the heart essentially tells the kidneys, I'm drowning up here, open the floodgates and let some fluid out. That elegant push and pull is how a healthy body maintains a perfect hemodynamic equilibrium. But what happens when there is a typo in the structural blueprint from birth?
That brings us to the congenital vascular anomalies. While many are, harmless anatomical quirks, there are 3 specific anomalies that are high yield for pathology because their structural defects lead to catastrophic clinical outcomes. The first is the berry aneurysm.
These are terrifying. They look like tiny, thin-walled, blood-filled berries hanging off the arteries at the base of the brain, specifically at the major branch points within the circle of Willis.
To understand the pathology, you have to look at the mechanism of their formation. They don't just appear randomly. They occur specifically at arterial branch points because of a congenital attenuation or severe thinning of the arterial media.
The smooth muscle layer.
Right. During fetal development, the smooth muscle cells simply fail to form a continuous layer right where the artery bifurcates. This leaves a focal weak spot in the wall, composed only of intima and a fragile layer of adventitia.
Wow.
For years, the relentless pounding pressure of the arterial blood hammers against that microscopic weak point, slowly ballooning it outward.
And the clinical logic is just brutal. The patient usually has no idea it exists. It causes zero symptoms until the day the wall finally gives way. It spontaneously ruptures, releasing pressurized arterial blood directly into the subarachnoid space of the brain. It causes an agonizing thunderclap headache and is very often a fatal intracerebral hemorrhage.
And for your exam, you must lock in the genetic association. Baryaneurysms are highly associated with autosomal dominant polycystic kidney disease, or ADPKD.
Wait, really? So polycystic kidney disease is the big link?
Yes. If you're presented with a clinical vignette about a patient with a history of massive cyst-filled kidneys who suddenly drops dead from a brain bleed, the diagnosis is a ruptured baryaneurysm.
The second congenital glitch involves crossing the wires between the high-pressure and low-pressure systems, arteriovenous, or AV fistulas.
Right. An AV fistula is an abnormal, direct physical connection between an artery and a vein. It completely bypasses the intervening capillary bed.
So, using our earlier analogy, instead of the rushing mountain or river slowing down by flowing into the vast, shallow lake of the capillaries, the river just dumps directly at full speed and full pressure into the delicate venous storage units.
The mechanism of injury here is entirely hemodynamic. Veins are just not designed to handle pressurized arterial blood. The vein will undergo immense physical stress, dilating and becoming thick-walled. But the systemic clinical outcome depends on the size of the fistula.
If it's a massive AV fistula, you are shunting a huge volume of blood away from the tissues that need it directly back to the heart.
Exactly. This creates a massive problem for the left ventricle. Because so much blood is bypassing the peripheral resistance, the systemic blood pressure drops. To compensate, the heart has to pump frantically, increasing both heart rate and stroke volume, desperately trying to maintain perfusion to the rest of the body.
And if the fistula is large enough, this chronic massive volume overload forces the heart to work itself to death, resulting in high-output cardiac failure.
Exactly right. Which brings us to the third anomaly, and this one is a beautiful illustration of how structural pathology triggers a systemic cascade: fibromuscular dysplasia, or FMD.
Okay, let's break this down. Demographically, FMD presents primarily in women, with a median age of diagnosis around 52. Morphologically, it is defined by focal irregular thickening of the walls of medium and large muscular arteries, most famously the renal artery supplying the kidneys.
Yeah.
But what exactly is thickening? Like, is it plaque?
No, it is not atherosclerosis. It is a non-inflammatory hyperplasia, an abnormal cellular overgrowth and fibrosis involving the media and intima layers. The smooth muscle cells and fibroblasts proliferate chaotically, creating these thick fibrotic ridges that protrude into the lumen of the artery, causing severe focal stenosis or narrowing.
And right next to these thickened ridges, the artery wall often becomes abnormally thin and attenuated, ballooning out slightly.
Which creates a highly characteristic appearance on an angiogram. The alternating areas of thick fibrotic ridges and thin balloon segments make the artery look like a string of pearls, and those attenuated outpouchings are structurally weak and are prone to rupture.
But the real diagnostic genius here is the clinical consequence. Like, how does a localized thickening in a single kidney artery trick the entire body into developing severe high blood pressure?
This is where we integrate the normal RAAS blueprint we built earlier. The physical narrowing of the renal artery in FMD acts like a kink in a garden hose. The blood flow dropping past that kink is severely reduced.
And sitting right past that kink are the juxtaglomerular cells, the kidney's pressure sensors.
Right, the sensors feel the low flow and low pressure. But the kidney doesn't know there's a localized physical blockage. The kidney assumes the pressure is low because the entire body is hemorrhaging or severely dehydrated. It basically hits the panic button,
It dumps massive amounts of renin into the blood.
triggering the entire angiotensin-aldosterone cascade. Angiotensin II brutally constricts the arterioles body-wide, and aldosterone forces the healthy opposite kidney to hoard salt and water. The systemic blood pressure shoots up to dangerous levels. The localized structural defect in FMD has caused a severe case of secondary renovascular hypertension.
Wow. The kidney is essentially destroying the entire cardiovascular system to save its own filtration rate. It's a perfect example of a biological system working exactly as designed but reacting to false information.
Which provides the perfect logical bridge to our next major topic: We've seen how a specific structural glitch causes secondary hypertension. But what happens when high blood pressure isn't a secondary response to a stricture, but a primary idiopathic disease?
That is essential hypertension. It accounts for roughly 95% of all clinical hypertension cases. Essential or primary just means idiopathic. We cannot point to a single tumor, a single stricture like FMD, or a single genetic defect to blame it on.
It is an incredibly complex interplay of multiple genetic polymorphisms and environmental factors like stress, obesity, and dietary sodium. However, even without a single culprit, the core pathogenic mechanism, the underlying why of the disease, almost universally involves a subtle, chronic defect in renal sodium excretion.
Let's break down that mechanism. In a perfectly healthy person with normal kidneys, if blood pressure rises, say, after eating a massive salty meal, the kidneys simply respond by filtering and peeing out the extra sodium and water. This is called pressure diuresis. The volume drops, and the pressure returns to normal.
But in essential hypertension, that elegant pressure diuresis mechanism is blunted. The kidneys become stubborn. At a normal arterial blood pressure, they simply refuse to excrete the body's daily sodium load.
So the sodium builds up in the blood, and where sodium goes, water follows, the blood volume expands.
Right. And to force these stubborn kidneys to finally excrete that sodium, the body has to drive the systemic blood pressure higher. The expanded volume and increased cardiac output push the pressure up until it reaches a new elevated threshold where the kidney is finally forced to filter the sodium.
The body has achieved a new steady state. The sodium is finally being managed.
But the biological cost is devastating. The entire vascular system is now subjected to chronic, relentless, high-pressure mechanical trauma. Over years and decades, this chronic hammering destroys the vessel walls.
Before we analyze that chronic microscopic destruction, we must define the acute medical emergency known as malignant hypertension. This occurs in about 5% of hypertensive patients, and it is catastrophic.
Malignant hypertension is defined by a rapidly rising, severe spike in blood pressure. We are talking about systolic pressures soaring over 180, or diastolic pressures over 120 millimeters of mercury.
The pressure is so high it literally causes the tiny vessels in the back of the eye to burst, resulting in retinal hemorrhages, and it causes the optic disc to physically swell, a condition called papilledema.
It causes acute, catastrophic renal failure. If left untreated, malignant hypertension is usually fatal within 1 to 2 years.
To understand why it's fatal and to dominate your pathology slide exams, you must absolutely master the histology of how high blood pressure damages the small blood vessels. This is the pathology of arteriolosclerosis, and there are two distinctly different microscopic forms you need to know: hyaline and hyperplastic.
Let's start with hyaline arteriolosclerosis. This is the morphological hallmark of chronic, benign, essential hypertension. It's the slow burn.
Okay, slide time. If you are looking at a slide of a kidney from a patient with chronic hypertension, you will see arterioles with walls that are intensely thickened by a homogeneous, structureless, glassy, pink staining material. That is the hyaline. The underlying cellular detail is completely obscured, and the lumen, the space where the blood flows, is severely narrowed.
The mechanism behind this pink appearance is crucial. Think of the arteriole like a concrete, seedy water pipe. For years, the water pressure inside the pipe has been slightly too high. This chronic mechanical stress injures the delicate endothelial cells lining the inside.
The nonstick coating gets damaged.
Exactly, it becomes leaky. Plasma proteins from the blood seep through the damaged endothelium and get physically trapped in the vessel wall. In response to this constant hemodynamic stress and the irritating presence of plasma proteins, the smooth muscle cells in the media react by churning out massive amounts of excess extracellular matrix.
So using the pipe analogy, the city responds to the leaking water by continuously pumping wet cement into the walls of the pipe. Eventually, the mixture of trapped plasma proteins and thick layers of extracellular matrix turns the entire pipe into a rigid, thick, glassy, pink block.
The structural consequence is that the vessel becomes stiff and the lumen becomes incredibly narrow. The blood simply can't get through. In the kidneys, this diffuse arteriolar narrowing starves the filtering units, the glomeruli of blood and oxygen. They slowly die and turn into scar tissue, a condition called nephrosclerosis.
And here is a massive conceptual link for your exam. You do not only see this pink hyaline arteriolosclerosis in hypertension, you see the exact same glassy pink vessels in diabetic microangiopathy.
Yes, the histology is identical, but the mechanism of injury is different. In diabetes, it isn't the mechanical trauma of high pressure causing the damage. It is the chemical trauma of chronic hyperglycemia. The high blood sugar directly damages the endothelial cells, causing the exact same plasma leakage and reactive matrix deposition.
That is the slow burn of hyaline. Now, contrast that with the explosive damage of hyperplastic arteriolosclerosis. This is the hallmark of severe malignant hypertension.
When you look at hyperplastic arteriolosclerosis under the microscope, the wall doesn't look like pink glass. It looks incredibly cellular. You see concentric, laminated rings of thickened tissue wrapping around the lumen, creating a classic onion skin appearance.
Why does it look like an onion? What is the vessel actually doing?
It is a frantic, desperate reaction to an acute, massive spike in blood pressure. The mechanical force is so severe that the smooth muscle cells in the vessel wall begin to rapidly proliferate. They multiply and secrete layers upon layers of thickened, reduplicated basement membranes, desperately trying to build concentric walls of armor to stop the vessel from rupturing.
But the terrible irony is that by building all those thick onion skin layers of armor inward, they completely obliterate their own lumen. The blood flow stops entirely. So is the vessel literally choking itself to death to save itself?
Essentially, yes. And because the pressure attack is so violent, this hyperplastic thickening is frequently accompanied by a specific type of cell death called fibrinoid necrosis. The sheer mechanical trauma literally tears the vessel wall apart. Smooth muscle cells die, the endothelium shatters, and massive amounts of fibrin from the blood leak into the dying tissue, creating a smudgy, intensely pink necrotic graveyard within the vessel wall.
The vessel explodes from the pressure and then tries to patch the crater with frantic onion skin scarring. It beautifully, tragically illustrates how hypertension is a systemic destroyer of small vessels. But hypertension doesn't stop at the small vessels. The relentless pressure also actively accelerates the decay of the large muscular arteries, setting the stage for our next major category.
Which brings us to the umbrella term, arteriosclerosis.
Arteriosclerosis is a generic word. It literally translates to hardening of the arteries. It is not one single disease entity. It is a category that encompasses 4 distinct pathological patterns.
The first pattern is arteriosclerosis, which we just thoroughly covered, the hyaline and hyperplastic diseases of the microscopic arterioles.
The second pattern is atherosclerosis, which involves the formation of massive lipid-filled plaques in the large arteries. This is the undisputed main event, and we are going to tear its molecular mechanisms apart in a moment.
The third pattern is a brief honorable mention for your studies. Fibromuscular intimal thickening, which is simply a general non-specific healing response where the vessel wall thickens after any kind of mechanical or inflammatory injury.
But the fourth pattern is a classic trap on pathology slide exams, Munkeberg medial sclerosis.
Munkeberg is characterized by the formation of ring-like calcific deposits, specifically within the media of medium-sized muscular arteries. When you look at a slide, you'll see these striking, amorphous, purplish-blue deposits because calcium stains dark blue on an H&E stain.
But the crucial diagnostic feature is location. Where exactly are these blue rocks sitting?
They are centered strictly on the internal elastic lamina. They form distinct, hard rings within the muscular wall of the artery.
So if I'm a medical student looking at a slide, and I see a vessel wall packed with terrifying dark blue calcifications, my instinct is to scream, atherosclerosis. The patient is having a massive heart attack.
But the clinical logic of Munkeberg is completely different, isn't it?
It is completely different because of one critical structural fact. Unlike an atherosclerotic plaque, the calcification in Munkeberg medial sclerosis does not encroach upon the vessel lumen.
The pipe gets stiff, but the opening stays exactly the same size.
Precisely. It makes the artery rigid and pulseless in severe cases, but it does not block the flow of blood. Therefore, it is generally considered clinically insignificant. It does not cause ischemia. It does not cause heart attacks.
It's an incidental finding. The text notes you frequently see it in patients over 50, and it often shows up on routine mammograms as striking, calcified blood vessels weaving through the breast tissue. It is a distractor on the exam. They will show you a heavily calcified vessel, the lumen will be perfectly open, and they will ask you what symptoms the patient is experiencing. The answer is none.
Got it. But now, we must face the true killer. The disease that causes roughly half of all mortality in the Western world, atherosclerosis.
For decades, people thought of atherosclerosis simply as grease clogging a pipe, a passive buildup of fat. But modern pathology understands it as something much more dynamic and sinister. The core pathogenesis of atherosclerosis is governed by the response to injury hypothesis.
This hypothesis states that a plaque is not just a passive lump of fat. It is a misguided chronic inflammatory healing process. It is a biological wound that cannot heal. Let's break down the exact step-by-step molecular chain reaction because this sequence is the foundation of cardiovascular pathology.
Step 1 is endothelial injury and dysfunction. In a perfectly healthy state, your endothelial cells form a pristine, non-stick, Teflon-like barrier. They secrete molecules that actively repel platelets and force the vessel to dilate. But the chronic exposure to stressors begins to break this non-stick surface down.
What kind of stressors?
The mechanical shear stress of hypertension constantly ripping at the cells, the chemical toxicity of thousands of compounds in cigarette smoke, the inflammatory presence of chronically high blood sugar and diabetes, or high levels of circulating lipids.
But these stressors don't necessarily strip the cells off the wall. The cells don't just peel away, they become dysfunctional.
Exactly. Their internal gene expression fundamentally shifts. They stop producing the nonstick molecules and instead begin expressing specialized adhesion molecules on their luminal surface, like microscopic strips of Velcro. At the same time, the tight junctions between the endothelial cells weaken. The barrier becomes leaky.
Which brings us to step 2, hemodynamic localization. This is a fascinating structural question. If I have high cholesterol, that cholesterol is bathing every single artery in my body. So why don't plaques form in a perfectly even layer everywhere? Why are they so patchy and localized?
Because the dysfunction we just talked about is heavily influenced by fluid dynamics. The source text explicitly notes that plaques predominantly form at the ostia of exiting vessels, at major branch points, and along the posterior wall of the abdominal aorta.
Why those specific anatomical spots?
Because those are areas of turbulent, chaotic blood flow. When blood hits a branch point, it swirls, creating irregular shear stress that physically traumatizes the endothelium, pushing it toward that dysfunctional Velcro-expressing state.
And the counterpoint is even more amazing. In the straight sections of your arteries where the blood flow is perfectly smooth and laminar, the physical friction of that smooth flow actually forces the endothelial cells to turn on specific atheroprotective genes.
Right. Smooth laminar flow actively signals the cells to defend themselves against inflammation. Turbulence silences those defenses.
Step 3 is where the biological poison enters the wall, lipid accumulation and oxidation. The endothelium is now leaky and dysfunctional. Low-density lipoprotein, or LDL, the classic bad cholesterol, slips out of the bloodstream, passes through the leaky endothelial gaps, and becomes physically trapped in the intima layer of the artery wall.
But simply having LDL trapped in the wall isn't what triggers a massive immune response. The trap is just the beginning. Once stuck in the intima, the LDL particles are exposed to the local microenvironment,
which is full of oxygen-free radicals, or reactive oxygen species, being produced by the struggling endothelial cells and local immune cells.
These free radicals chemically attack the trapped LDL, mutating it into oxidized LDL. And oxidized LDL is an incredibly toxic, highly inflammatory molecule. It directly injures neighboring cells, triggering them to release even more chemical distress signals. Furthermore, those same oxygen-free radicals actively destroy nitric oxide.
And nitric oxide is the molecule that normally tells the smooth muscle cells to relax and the vessel to dilate. So by destroying it, the free radicals force the vessel into a stiff, constricted, highly irritable state.
This toxic environment sets off step 4, foam cell formation. The endothelial cells, panicking from the toxic oxidized LDL, use their newly expressed Velcro adhesion molecules to catch passing monocytes, a type of white blood cell, out of the bloodstream.
They pull the monocytes out of the blood and drag them down into the intima.
Once inside the wall, the monocytes transform into active macrophages. Their primary instinct is to clean up a mess. They recognize the oxidized LDL as toxic waste, and they attempt to clear it out by eating it via specialized scavenger receptors on their surface, like SRA and CD36.
But there is a fatal flaw in this cleanup operation, isn't there?
Yes. Normal cellular receptors for LDL have a negative feedback loop. When the cell has enough fat, it stops eating. But these scavenger receptors have no off switch. The macrophages simply eat and eat and eat, gorging themselves on the toxic oxidized fat until their cytoplasm is utterly bloated with massive lipid droplets.
They become so stuffed with fat that under a microscope they look like bubbling, frothy spheres. They become foam cells. And because they can't actually digest the oxidized lipid, they eventually undergo apoptosis. They burst and die, spilling their toxic fatty guts back into the tissue, creating a necrotic core of dead cells and fat.
Which forces the vessel to initiate step 5, drastically altering its physical structure. Smooth muscle cell recruitment. The dying foam cells, the traumatized endothelium, and any platelets that have stuck to the leaky wall all start screaming for help. They release powerful chemical distress signals, primarily platelet-derived growth factor, or PDGF, and fibroblast growth factor, FGF.
And who receives that signal? The smooth muscle cells sitting in the media layer below. But wait, I want to make sure I understand the gravity of this. We established earlier that smooth muscle cells exist solely to contract and regulate blood pressure. They are muscle. But these signals cause them to fundamentally change their biological identity.
It is a profound phenotypic switch. In response to EDGF, the smooth muscle cells essentially forget they are muscle. They stop being contractile cells. They degrade the elastic membrane separating them from the intima, physically migrate upward into the growing lesion, and transform into synthetic cells.
They start acting like fibroblasts, scar tissue builders.
Exactly, which is step 6, extracellular matrix synthesis. These recruited, transformed smooth muscle cells rapidly proliferate inside the intima. They begin furiously pumping out massive amounts of collagen, elastin, and proteoglycans.
What are they trying to achieve?
They're trying to heal the wound. They're attempting to build a thick, dense, fibrous cap over the top of the toxic, inflammatory, fatty mess, trying to wall it off from the bloodstream, much like building a scar over a cut on your skin.
But here's the ultimate tragedy of the disease. A cut on your skin heals because the splinter is removed, the bacteria are killed, and the injury stops. But in atherosclerosis, the injury never stops.
Precisely, because the patient usually has chronic hyperlipidemia, high cholesterol, new LDL particles continuously leak into the wall. The toxic oxidized lipids are constantly replenished. New macrophages rush in, gorge themselves, and die. The smooth muscle cells desperately try to keep building the scar, but the inflammatory fire underneath keeps expanding. The body traps itself in a vicious, never-ending cycle of injury, inflammation, and frantic, failing repair.
And that continuously growing lump of fat, dead cells, and thick collagen scarring is the atherosclerotic plaque. That intricate failed healing mechanism is the perfect bridge to Section 6, integration with immunology and risk factors.
To fully understand cardiovascular risk, we have to look beyond just the structural biology and understand the epidemiological risk factors that drive this inflammatory cycle. We know the classic modifiable risk factors: hyperlipidemia, hypertension, smoking, and diabetes.
The source material makes a critical point about how these risk factors interact. They don't just add together, they multiply. If you have 2 risk factors, your risk of a myocardial infarction doesn't double. It increases roughly 4-fold. If you have 3 risk factors, for example, a patient who smokes, has uncontrolled hypertension, and high cholesterol, their risk of a fatal event increases 7-fold compared to a healthy baseline.
Let's focus on the hyperlipidemia, because the type of cholesterol matters intensely. LDL low-density lipoprotein is the primary villain. It is the delivery vehicle that transports cholesterol from the liver out to the peripheral tissues and, crucially, delivers it straight into the artery walls.
While HDL high-density lipoprotein is the hero of the story.
Right. HDL is the cleanup crew. It participates in reverse cholesterol transport. It can physically extract cholesterol out of the existing foam cells within the plaque and transport it back to the liver for excretion in the bile. Higher levels of HDL directly correlate with lower cardiovascular risk.
This balance between LDL and HDL is why statins are arguably the most revolutionary drugs in cardiovascular medicine.
Statins are a pharmacological triumph. Their primary mechanism is inhibiting HMG-CoA reductase, which is the key rate-limiting enzyme the liver uses to synthesize its own internal cholesterol.
So if you block that enzyme, the liver panics because it needs cholesterol to function. Where does it get it?
It creates more LDL receptors on its surface and aggressively pulls LDL out of the circulating bloodstream, drastically lowering the patient's serum LDL levels. But statins are brilliant because they also have pleiotropic effects, secondary benefits independent of just lowering cholesterol. Statins directly reduce plaque inflammation, improve endothelial function, and help stabilize the fibrous cap, making the plaque less likely to rupture.
We also have the non-modifiable risks: age, genetics, and gender. The text notes a fascinating gender disparity. Premenopausal women are remarkably protected against atherosclerosis compared to men of the same age.
The assumption for decades was that estrogen was directly protective.
But biology is rarely that simple. The text specifically highlights that when large clinical trials gave postmenopausal women estrogen replacement therapy, assuming it would protect their hearts, it didn't work. In fact, it slightly increased their overall cardiovascular risk. It remains a complex endocrine paradox.
But the truly cutting-edge area of cardiovascular pathology, the area where the textbooks are literally being rewritten right now, is the deep integration of atherosclerosis with the innate immune system. Atherosclerosis is no longer viewed just as a lipid disease. It is an immune disease.
This brings us to the molecular machinery of the inflammasome. We talked about macrophages eating the oxidized LDL and turning into foam cells. But they don't just eat fat droplets. They also engulf solid, jagged crystals of cholesterol that form inside the necrotic core.
And this is where the immune system goes nuclear. When a macrophage ingests these sharp cholesterol crystals, the crystals cause physical damage inside the cell. This damage activates specialized cytosolic innate immune receptor sensors that usually exist to detect bacterial toxins or viral DNA.
But here, they are being triggered by the body's own crystallized cholesterol.
Once triggered, these sensors assemble into a massive multi-protein complex called the inflammasome. The inflammasome's primary job is to activate an enzyme called caspase-1, which then takes an inactive precursor protein and chops it into interleukin-1 beta, or IL-1.
And interleukin-1 beta is one of the most potent, aggressive, pro-inflammatory cytokines in the entire human body.
It floods the local tissue. It recruits massive numbers of T lymphocytes and fresh monocytes into the plaque, pouring gasoline on the inflammatory fire, accelerating the tissue destruction and plaque growth.
This isn't just theory. The clinical proof is astounding. The source references trials, like the famous CANTOS trial, showing that if you give high-risk cardiovascular patients a monoclonal antibody drug that specifically neutralizes interleukin-1 beta, you significantly reduce their rate of recurrent, non-fatal myocardial infarctions.
The patient's cholesterol levels remain exactly the same. The fat is still there. But simply by blocking the immune system's inflammatory response to that fat, the progression of the disease is halted. It proves definitively that the inflammation is driving the lethality of the disease.
This immune revelation also changes how we test for cardiovascular risk. Because interleukin-1 and other cytokines are flooding the system, they eventually reach the liver. And the liver responds to these inflammatory alarms by synthesizing C-reactive protein, or CRP.
CRP is an incredibly sensitive, easily measured blood marker for systemic inflammation. Historically, it was used to track infections or rheumatoid arthritis. But now, high-sensitivity CRP assays are used to independently predict the risk of myocardial infarction and stroke.
Even in people who appear perfectly healthy and have normal cholesterol levels, if their CRP is elevated, it means that hidden inflammatory fire is burning inside their blood vessels, and their risk of a sudden plaque rupture is significantly higher.
Beyond the inflammasome, your professors will absolutely test you on two specific, relatively newly understood risk mechanisms. The first is a unique genetic variant called lipoprotein A, or LPA.
Let's break down why this specific particle is so dangerous. It's essentially an LDL particle, so it carries all the bad cholesterol, but it has a massive extra protein looped around it called apolipoprotein. Why does that extra protein turn it into a double threat?
Because of molecular mimicry. That extra apolipoprotein is structurally nearly identical to plasminogen, the molecule the body uses to dissolve and break down blood clots. Because they look so similar, LPA competitively inhibits plasminogen activation.
So it binds to the sites where plasminogen is supposed to work, but it can't actually dissolve the clot.
Exactly. So LPA is actively driving the lipid accumulation to build the plaque and simultaneously preventing the body from breaking down fatal blood clots that form on top of the plaque. It's a profoundly atherogenic molecule.
Wow. And the second new mechanism is truly wild, bridging the gap between hematology, aging, and cardiology. Clonal hematopoiesis of indeterminate potential, or CAP.
This requires a deep dive into stem cell biology. Deep inside your bone marrow, you have hematopoietic stem cells constantly dividing to create all your white and red blood cells. As we age, it is common for a single one of these stem cells to acquire a random somatic mutation, often in epigenetic regulator genes like TET2 or DNMT3A.
This isn't a genetic mutation you inherit from your parents. It's an acquired error in one specific cell.
Crucially, this mutation gives that single stem cell a survival and proliferation advantage over its healthy neighbors. It begins dividing faster, creating a massive clone army of daughter white blood cells that all carry the mutation.
But let me clarify for the listener, these patients do not have leukemia. Their total white blood cell count is completely normal.
They appear hematologically perfectly healthy. But epidemiologists made a staggering discovery. Older individuals harboring these CHE mutations have a drastically disproportionately increased risk of dying from cardiovascular disease. Why? Because the mature monocytes generated by this mutated clone army have fundamentally altered innate immune functions.
When these mutated trigger-happy monocytes enter the artery wall and become macrophages, they are hyperinflammatory. They secrete massive amounts of IL-1 and other cytokines, accelerating atherogenesis at a terrifying rate.
Your bone marrow mutates, and your heart pays the ultimate price decades later. The immune system is the architect of the plaque's destruction.
Which means your blood vessels are essentially keeping a biological diary of your hemodynamics, your diet, and your immune system's mutations. Let's look at the actual pages of that diary. What do these lesions physically look like under the microscope? Section 7. Atherosclerosis morphology, histology, and clinical logic.
The very first visible entry in that biological diary is the fatty streak. Morphologically, if you open up an artery, you will see minute, flat, yellow macules. Over time, these tiny spots can coalesce into longer, elongated yellow streaks running along the vessel wall.
Histologically, what is actually in a fatty streak? Is it a full plaque?
No, it is the earliest precursor. A fatty streak is composed almost entirely of those lipid-filled, foamy macrophages sitting in the intima. There is no significant necrotic core yet, no massive smooth muscle proliferation, and no thick fibrous cap. Crucially, because they are flat, they do not protrude into the lumen. They cause zero flow disturbance and zero symptoms.
And here's the most mind-blowing high-yield fact from the source text regarding the timeline of this disease. Fatty streaks are present in the aortas of infants younger than 1 year old, and they are found in virtually all children older than 10 years.
Regardless of their genetics, geography, or diet, it suggests that this initial trapping of lipid and macrophage response is a fundamental, almost unavoidable consequence of human vascular hemodynamics.
Now, not every fatty streak progresses to a deadly plaque, but the plaques that kill adults form in the exact same anatomical locations where those fatty streaks appeared when they were teenagers. The biological diary started writing itself in adolescence.
Over decades, driven by those risk factors we discussed, some of those streaks evolve into the fully mature atherosclerotic plaque. Morphologically, these are described as eccentric, patchy lesions.
Eccentric, meaning they usually don't form a perfect ring around the entire vessel. They grow on one side of the wall, bulging inward, creating an off-center, narrowed lumen.
Let's dissect the microscopic architecture of a mature plaque, starting from the blood flowing in the lumen and moving deep into the vessel wall. The roof of the lesion is the fibrous cap.
This is the scar tissue we talked about earlier. It's built by those recruited smooth muscle cells and is made of dense collagen and elastin, attempting to wall off the danger.
At the edges of this cap, where it slopes down to meet this relatively normal vessel wall, is an area called the shoulder. The shoulder is critical because it is the most highly cellular area of the plaque. It is packed with active macrophages, T cells, and smooth muscle cells. It is the active, ongoing battleground where the inflammation is hottest.
And buried deep beneath the fibrous cap, down in the intima, is the necrotic core.
This is the graveyard. It is a disorganized mass of extracellular lipid, dead and dying foam cells, fibrin, and necrotic cellular debris.
And when you look at this necrotic core on a histology slide, you will see a very specific classic feature, cholesterol clefts. They look like sharp, empty, white, needle-shaped spaces scattered throughout the pink and purple debris. Why are they empty?
That is a classic pathology artifact. In the living patient, those spaces were filled with solid, jagged, highly inflammatory crystals of cholesterol. But to make a slide, pathologists have to wash the tissue in chemical solvents like alcohol and xylene to preserve it. These organic solvents literally dissolve and wash away all the crystalline lipid, leaving behind only the empty, needle-shaped tracts where the crystals used to be.
It's the footprint of the fat. Another key histological feature you'll see in large plaques is neovascularization.
The plaque becomes so massively thick that oxygen from the main arterial lumen can't diffuse all the way through it. The cells inside the plaque begin to starve. In response, tiny, fragile new blood vessels sprout from the vasa vasorum and the adventitia and grow directly into the plaque to supply it with blood.
And those fragile new vessels are going to play a fatal role in a moment. But first, let's look at the clinical logic. How does this structure actually cause disease? The first mechanism is atherosclerotic stenosis.
This is the slow, chronic consequence. As the plaque grows, the vessel initially tries to adapt by remodeling outward, expanding its outer wall to keep the lumen open. But eventually the plaque grows so massive it pushes inward, physically narrowing the lumen and restricting blood flow.
The crucial clinical tipping point for the exam is 70% occlusion. That is defined as critical stenosis.
At 70% occlusion, a fascinating clinical threshold is crossed. While the patient is resting, sitting on the couch, the restricted blood flow is still just enough to meet the heart muscle's low demand for oxygen. They feel perfectly fine.
But the second they exert themselves, they walk up a flight of stairs or run for a bus. The heart's demand for oxygen spikes. The stenotic artery simply cannot deliver enough blood past the 70% blockage. The heart muscle becomes ischemic and screams in pain. This manifests as stable, angina-predictable, reproducible chest pain on exertion.
But stable angina, while painful, is not what usually kills people suddenly. Sudden catastrophic cardiac death is driven by acute plaque change.
The plaque suddenly fails. It stops being a slow, chronic blockage and becomes an immediate, explosive emergency. The source text details 3 specific mechanisms of acute plaque change.
The first and most common is rupture or fissuring of the fibrous cap. The physical stress of the blood pressure, combined with enzymes chewing away the collagen, causes the fibrous cap to suddenly tear open.
The second the cap tears, the highly thrombogenic, highly toxic necrotic core is exposed directly to the flowing blood. The blood instantly recognizes it as a massive injury and triggers a massive coagulation cascade. A blood clot, or thrombus, forms instantly, completely occluding the artery.
The second mechanism is erosion or ulceration. The endothelial cells covering the plaque simply peel away or die, exposing the underlying subendothelial basement membrane. This too triggers immediate platelet adhesion and fatal thrombosis, even without a deep rupture into the core.
And the third mechanism involves those tiny new blood vessels we just mentioned, hemorrhage into the plaque.
Those neovascular sprouts are incredibly fragile. If the high systemic blood pressure causes one of those tiny vessels to burst inside the plaque, it creates an expanding hematoma, a balloon of blood inflating inside the confined space of the lesion. This sudden internal pressure can cause the plaque to rapidly expand and block the artery, or it can generate enough force from the inside to blow out the fibrous cap.
I like to visualize this using a jelly donut analogy. The necrotic core is the toxic lipid jelly, and the fibrous cap is the doughy crust holding it in. Based on the structure, we can classify plaques as either stable or vulnerable.
A stable plaque is a donut with an incredibly thick, dense collagen crust, a relatively small lipid core, and very minimal inflammation in the shoulder. It might slowly grow and cause that predictable 70% critical stenosis and stable angina, but its thick crust makes it highly unlikely to suddenly pop.
Contrast that with a vulnerable plaque. This is a jelly donut with a terrifyingly thin, fragile crust, a massive, bulging lipid core, and shoulder regions completely packed with inflammatory cells.
And those inflammatory macrophages are actively secreting matrix metalloproteases, or MMPs, enzymes that are literally digesting the collagen in the thin cap, making it weaker by the hour.
It is a microscopic time bomb waiting for the slightest pressure spike to explode. And this vulnerability perfectly explains one of the most famous, chilling clinical statistics in cardiology, the circadian rhythm of heart attacks. Have you ever noticed that the incidence of acute myocardial infarctions heavily peaks in the morning, between 6 a.m. and noon?
It is entirely driven by the waking adrenergic surge. When you wake up, get out of bed, and start your day, your sympathetic nervous system kicks in. You release cortisol and catecholamines. Your blood pressure suddenly spikes, your heart rate increases, and your blood platelets become stickier and more reactive.
That sudden morning spike in mechanical stress, that sudden forceful squeeze on the artery walls, is very often the physical trigger that finally snaps the weakened crust of a vulnerable plaque. The cap tears, the toxic lipid core is exposed, a massive thrombus forms in seconds, and the patient suffers a fatal morning heart attack.
It is the devastating culmination of decades of silent inflammation. But plaques and occlusions aren't the only way a vessel can fail. Sometimes the injury doesn't cause the vessel to narrow. It causes the wall to completely lose its structural integrity. Which brings us to our final dramatic chapter, Section 8, Aneurysms and Dissections.
When the walls simply give way. An aneurysm is defined as a localized, abnormal dilation of a blood vessel or the heart. But we have to be precise with our terminology. We differentiate between true aneurysms and false aneurysms.
A true aneurysm means the structural integrity of the wall has weakened, but the wall itself is still intact. All 3 layers, the intima, the media, and the adventitia balloon outward together. The blood remains safely inside the confines of the stretched vessel wall.
A false aneurysm, or pseudoaneurysm, is much more violent. It is actually a physical hole, a full-thickness defect in the vessel wall.
The blood escapes through the hole, But instead of bleeding freely into the body cavity, the escaping blood is contained by a layer of surrounding external connective tissue or by a massive organized hematoma. It creates a pulsating sac of blood sitting completely outside the actual artery, communicating with the lumen through the hole.
Morphologically, when surgeons or pathologists describe aneurysms, they classify them by shape. They are either saccular or fusiform.
A saccular aneurysm is a discrete, spherical outpouching involving only a portion of the vessel's circumference. It looks like a bubble forming on one side of tire. They typically range from 5 to 20 centimeters in diameter and are often partially filled with clotted blood.
A fusiform aneurysm is a symmetrical circumferential dilation. The entire segment of the artery balloons outward evenly like a snake that swallowed a rat. They are very common in the aortic arch and abdominal aorta.
But this shape is just descriptive. The critical question for your pathology seminar is the pathogenesis, the why. Why does the thick, muscular, incredibly strong wall of the aorta weaken so drastically that it balloons outward?
It all comes down to a critical, catastrophic imbalance of extracellular matrix. The vessel wall loses its tensile strength, the synthesis of new collagen and elastin fails, or the degradation of existing matrix accelerates. And there are three main pathways that drive this destruction. Let's start with genetics.
The classic genetic pathway involves mutations in the TGF-beta signaling pathway, most famously seen in Marfan syndrome.
Let's break down the exact molecular mechanism of Marfan, because this is a guaranteed exam question. Patients with Marfan syndrome have a mutation in the gene encoding a structural scaffold protein called fibrillin-1.
In a normal, healthy aorta, fibrillin acts as a physical scaffold, but it also has a crucial regulatory role. It physically binds to and sequesters TGF-beta, keeping it inactive. Because the fibrillin is defective in Marfan patients, it cannot hold on to the TGF-beta.
So the TGF-beta floats free, becoming excessively bioavailable in the aortic wall.
And this overactive TGF-beta signaling essentially corrupts the smooth muscle cells. It alters their gene expression, leading to inadequate, defective synthesis of elastic tissue and an overproduction of amorphous, gooey ground substance. The aorta loses its strong rubber band snap. It just stretches, weakens, and dilates.
The second pathway to aneurysm formation is enzymatic destruction. We saw this in the atherosclerotic plaques.
The massive army of macrophages recruited into the atherosclerotic plaque doesn't just eat fat. They release matrix metalloproteases, or MMPs. These are literal tissue-digesting enzymes. They chew up and degrade the elastin and collagen in the underlying arterial media, physically destroying the structural integrity of the wall.
And the third pathway is ischemia. The wall of the aorta physically starves to death.
How does the aorta, a massive pipe carrying gallons of highly oxygenated blood, suffer from lack of oxygen? It's a problem of diffusion distance. The inner third of the aortic wall gets its oxygen directly from the blood rushing past in the lumen. But if you have a massive, thick atherosclerotic plaque sitting on the intima, it dramatically increases the physical distance the oxygen has to travel.
Yeah.
The inner media starves.
What about the outer media?
The outer media relies on its own dedicated network of microscopic blood vessels called the vasa vasorum, the vessels of the vessels. But in patients with severe systemic hypertension, those tiny arterioles of the vasa vasorum undergo the exact same hyaline arteriolosclerosis we discussed earlier. Their walls thicken, their lumens narrow, and blood flow to the outer aorta is choked off. The outer media starves too.
The smooth muscle cells in the media, caught between a thick plaque and narrowed vasa vasorum, slowly die. And when we look at this ischemic destruction under the microscope, we see a highly specific histological pattern called cystic medial degeneration.
The normal aortic media looks like perfectly layered lasagna, neat parallel layers of elastic tissue and smooth muscle. But in cystic medial degeneration, that structure is obliterated. You see massive dropout and death of the smooth muscle cells. The elastic fibers are shattered and fragmented, and the empty spaces are filled with large amorphous pools of pale blue-staining proteoglycans.
And here is a massive memory tip for the exam. The name is cystic medial degeneration, but despite the name, there are no true cysts in the tissue.
It is entirely a descriptive term. The pools of blue proteoglycans look like empty cystic spaces under low magnification, but they are not true epithelial-lined cysts. Do not fall for a trick question claiming they are true cysts.
Now let's look at the two major clinical manifestations of this wall destruction. First, the abdominal aortic aneurysm, or AAA.
Location is the defining feature here. AAAs almost universally occur below the branch point of the renal arteries and above the bifurcation of the iliac arteries. Who gets them? typically men over the age of 50, and smoking is a massive independent risk factor.
And the primary driving force behind almost all AAAs is severe, complicated atherosclerosis. The massive plaques thin and destroy the underlying media through those MMP enzymes and ischemic mechanisms we just described.
These aneurysms can grow to staggering sizes, up to 15 centimeters in diameter, and clinically, when you look inside one, it is almost always filled with a thick, laminated, poorly organized blood clot called a mural thrombus. The altered, swirling blood flow inside the dilated sac triggers chronic clotting.
While atherosclerosis is the main cause, there are a few special variants of AAA you need to recognize. You have inflammatory AAAs, which occur in younger patients and are characterized by intense, dense periodic fibrosis and massive macrophage infiltration.
And within that inflammatory category is a highly specific subset, IgG4-related disease. This is a fascinating systemic autoimmune condition where multiple tissues, the pancreas, the salivary glands, and the aorta, are infiltrated by plasma cells, specifically expressing IgG4 antibodies. This causes intense tissue-destroying fibrosis.
The crucial clinical logic for the IgG4 variant is treatment. Unlike a standard atherosclerotic AAA, which requires surgery, the IgG4 variant is an immune disease and actually responds remarkably well to high-dose steroids and B-cell depletion therapy.
You also have mycotic aneurysms. These occur when circulating bacteria, for example, from an infected heart valve in endocarditis or salmonella from gastroenteritis, physically lodge in the aneurysm wall. The multiplying bacteria accelerate the destruction of the media and trigger rapid, fatal rupture.
But for a standard AAA, the clinical logic and surgical guidelines boil down to a single metric size.
The risk of an AAA rupturing is directly proportional to its diameter. An aneurysm less than 4 centimeters almost never ruptures, but the risk skyrockets exponentially when the aneurysm reaches 5.5 centimeters in diameter. At that point, surgical intervention with a graft is critical, because if a 6-centimeter aneurysm ruptures into the peritoneal cavity, the patient bleeds out internally and the resulting hemorrhage is almost always fatal.
Now let's move up the chest. Thoracic aortic aneurysms. These occur in the ascending aorta and the aortic arch, and they have a different set of primary causes. They are more frequently associated with chronic hypertension, Marfan syndrome, and bicuspid aortic valves.
And we absolutely must integrate microbiology here because historically a classic text cause of thoracic aneurysms was tertiary syphilis.
The Treponema pallidum spirochete. How does a sexually transmitted bacteria cause a massive aneurysm in the chest decades later?
It is a remarkable piece of pathogenesis. In the tertiary stage of the disease, the spirochetes have a bizarre predilection for infecting the vasorum, specifically the ascending aorta. The body mounts a massive cellular immune response to the bacteria, characterized by plasma cells. This intense inflammation causes obliterative endarteritis. It literally scars, thickens, and completely chokes off those tiny blood vessels,
Which causes severe ischemic injury to the outer media of the ascending aorta. The elastic tissue dies, the wall weakens, and it dilates into a massive aneurysm. The clinical signs of a thoracic aneurysm, regardless of cause, are all about compression of surrounding structures.
It pushes on the airway, causing respiratory distress. It compresses the esophagus, causing feeding difficulties. And as the aorta expands, it can stretch the recurrent laryngeal nerve looping around the arch, causing a persistent, unexplained cough or change in voice.
Which brings us to our final and undeniably most catastrophic vascular emergency, aortic dissection.
A dissection is fundamentally different from an aneurysm. It is not a ballooning of the wall. It is a violent, pressurized tearing apart of the wall.
The mechanism requires two sequential catastrophic events. First, there must be an intimal tear. The delicate inner lining of the aorta physically rips open. Second, the incredibly high-pressure systemic blood ejected from the left ventricle finds that tear, enters the vessel wall, and physically forces its way deep into the media layer.
It acts like a pressurized wedge. The blood violently unzips the aorta, splitting the media apart, usually right between the middle and outer thirds. As the blood pushes forward, it carves out a massive false channel running parallel to the true lumen of the artery.
Who is at risk for this? The epidemiology points to two main groups. First, men aged 40 to 60 with a long history of severe hypertension. As we discussed, the hypertension caused medial degeneration, making the wall structurally susceptible to tearing.
The second group comprises younger patients with connective tissue disorders, primarily Marfan syndrome. Their defective fibrillin and lack of elastic tissue make the aorta incredibly fragile.
And there is a rare but tragic third demographic, third trimester pregnancies. The intense systemic hormonal vascular remodeling required for pregnancy, combined with the extreme hemodynamic volume stress of the third trimester, can occasionally trigger a spontaneous catastrophic dissection in otherwise healthy women.
Morphologically, the initial intimal tear is usually transverse or oblique, and it most commonly occurs within 10 centimeters of the aortic valve, where the physical shear stress of the blood ejecting from the heart is highest. The dissecting hematoma then rushes downward towards the abdomen. Sometimes the blood will rip back through the intima further down the vessel, re-entering the main lumen and creating a functioning, double-barreled aorta.
Wait, there is a detail in the source text that completely blew my mind regarding dissections and atherosclerosis. We have spent this entire deep dive talking about how atherosclerosis is a vicious disease that destroys the structural integrity of the vessel wall, but the text states that the heavy, calcified scarring of severe atherosclerosis actually prevents an aortic dissection from spreading further down the vessel.
It is one of the darkest, most morbid ironies in all of human pathology.
Yes.
Aortic dissections are highly unusual in areas of the aorta with heavy, mature atherosclerotic plaque, because the massive medial fibrosis, the dense collagen, and the calcified scarring associated with a severe plaque essentially act like a physical brick wall inside the tissue. When the dissecting hematoma is tearing down the media, it hits that dense scar tissue and is physically arrested. It can't split the layers apart because the chronic inflammation of the plaque has glued the layers of the aorta inextricably together.
The plaque is so scarred and dead that it actually stops the tearing. Clinically, when a dissection occurs, it presents as the sudden, explosive onset of excruciating pain. Patients describe it as a tearing or stabbing sensation beginning in the anterior chest and rapidly radiating to the back, migrating downward between the shoulder blades as the dissection advances.
The surgical triage and the patient's chance of survival depends entirely on anatomical classification. The most widely used system divides them into type A and type B. Type A dissections are proximal. They involve the ascending aorta, regardless of how far down they travel. Type A is an immediate, catastrophic surgical emergency.
While type B dissections are distal, meaning they begin beyond the subclavian artery and only involve the descending aorta, they can often be managed medically with aggressive blood pressure control. But why is type A so incredibly lethal?
Because of anatomy. The ascending aorta is physically enclosed inside the pericardial sac, the tough, fibrous membrane surrounding the heart. The most common cause of death in a type A aortic dissection is not the patient bleeding out into the abdomen.
It's the dissecting blood tearing in the wrong direction.
Exactly. The high-pressure hematoma dissects backward, toward the heart, and ruptures outward through the adventitia. The blood instantly explodes into the confined space of the pericardial sac,
Causing massive cardiac tamponade. The pericardial sac fills with pressurized blood. It crushes the heart from the outside. The right ventricle simply cannot open to accept venous return. The cardiac output drops to zero, and the patient dies in moments.
And with that catastrophic microscopic tear, we have reached the end of our journey.
We have unpacked the entire vascular blueprint, from the elegant, perfectly regulating smooth muscle cells of a healthy arteriole, through the inflammatory battleground of the atherosclerotic plaque, all the way down to the lethal, unzipping mechanics of a type A dissection. You now possess the exact step-by-step logic, the cellular mechanisms, and the histological reality needed to absolutely dominate your pathology seminar.
You don't just know what the names of the diseases are. You understand the profound underlying structural why. You see the biological chain reactions that connect a tiny change in flow dynamics to a fatal anatomical rupture decades later.
Before we sign off, I want to leave you with one final, chilling reflection drawn directly from the text we analyzed. We talked extensively about fatty streaks, those flat, foamy yellow marks. The fatty streaks that will ultimately dictate the cardiovascular health and survival of a 60-year-old were already physically forming in the exact same anatomical branch points when they were a teenager.
Your blood vessels have been keeping a continuous, microscopic biological diary of every hemodynamic stress, every dietary choice, and every innate immune response since childhood. Every twist of the arterial nozzle, every wave of systemic inflammation is permanently recorded in the intima. It is a diary written relentlessly in lipid, macrophage debris, and collagen scar tissue.
So, as you head into your exam, think about that. The question isn't just what disease you will diagnose on a slide today. The question is, what chapter are your own vessels writing right now?